Module 8, Objective 5, describe the structures of the external, middle, and inner ear, explain their roles in equilibrium and hearing, and trace the pathways for equilibrium and hearing to their destinations within the brain. Here we have an image of the external, middle, and inner ear, and I know we cover these in lab to some degree, but it's just worth noting the external ear consists of largely this large flap of tissue that sits on the side of our head called the auricle, and its job is to capture sound waves, and as sound waves hit the side of the head, they swirl around within the whorls within the auricle, and they end up in the external acoustic meatus, which is then going to travel, and those sound waves are going to hit off the tympanic membrane, which separates the external from the middle ear. The middle ear, from the tympanic membrane, you have a series of bony ossicles. These are bones, the smallest bones within the body, and they include, in order, from the tympanic membrane, the malleus, right here, the incus, and then, of course, the stapes. And the stapes is going to interact with the first segment of the internal ear, which we'll note later. Additionally, the tympanic cavity gives rise to a tube called the auditory tube, which is connected to our nasopharynx, our throat. And then we have the internal ear, and the internal ear contains a couple structures. There's the cochlea, which is this snail shell-shaped structure. You have a large bulbous region here called the vestibule, and then we have the semicircular canals situated up here. So those constitute the anatomy of the external, middle, and inner ear. Here we see the organs of the inner ear. Again, the cochlea. Cochlea is responsible for the sensation of hearing. We have the vestibule right here. If you're going with the snail shell or the snail example, the vestibule would sort of be the head of the snail. And then the semicircular canals, of which there are three. They would be the antenna of the snail. And the vestibule and the semicircular canal are responsible for various aspects of our balance. So the vestibule encloses two chambers, the saccule and utricle, and receptors within these detect gravity and linear acceleration. To sort of simplify that, I always consider the vestibule associated with horizontal acceleration as well as vertical. And I just wrote those backwards. This is horizontal and this is vertical. If you're moving in a car, you're moving horizontally, and even with your eyes closed, you can sense that movement, and that's due to the vestibule. If you're moving up in an elevator, even though you don't see that you're moving, you can feel that due, again, to the sensation of the vertical acceleration from the vestibule. The semicircular canals have receptors that are stimulated by the rotation of the head, and they sort of sense circular rotation. If you were spinning in a chair, the semicircular canals would pick that up. And then lastly, the cochlea. It contains a cochlear duct, which we'll see has receptors that provide for sense of hearing. All of these structures are based on the same type of cell called the hair cell, and that's what we have right here, the hair cell. The hair cell is a specialized neuron that is going to generate excitement in the form of an action potential when stimulated. And usually it involves the bending of these hair-like structures that are at the apical end of this cell. And usually those hairs sort of project into some type of fluid, and when that fluid causes those hairs to bend, then it generates excitement, and you get the formation of an action potential, which will then be sent via the sensory nerve. So the hair cell is sort of the basic functional unit within all of these structures of the internal ear. Let's start with the semicircular ducts. There is an anterior, posterior, and lateral semicircular duct, and they're continuous with the utricle, one of the sacs within the vestibule. Each duct contains an expanded region called the ampulla, and it has a gelatinous mass that those hairs set in called the cupula. Each hair cell in the vestibular complex has about 100 stereocilia, and they kind of resemble long microvilli. In addition, they have one large kinocilium as well. This image just shows the relationship. Here we see the 3 semicircular canals and how they're continuous with the utricle. Here's the situation within the semicircular canals. You have your hair cells right here, and you can see that their hairs are embedded excuse me, in a very large sort of gelatinous paddle called the cupula. And that is within those tubes, those semicircular canals, filled with a fluid that is called endolymph. So here's the idea. If you were to start spinning in a chair, the fluid within these semicircular canals are going to also spin, and they're going to press on this cupula, and they're going to cause the cupula to bend. And as that cupula bends, these hair cells are going to bend, and they're going to get excited, and they're going to generate an action potential that is going to send a signal to the brain telling you that you're spinning. It's often fun to spin yourself in a chair and then stop the chair and then get up and try to run. And we all know the outcome of that. And what happens is, as you're spinning in that chair, all this fluid is moving with you, and it's causing that ampulla to bend, and it's causing those hair cells to bend, and it's sending a signal to your brain telling it that you're spinning. When you stop the individual or yourself quickly, while your body stops, this fluid continues to slosh, and it's continually sending signals to your brain. So your brain's getting mixed signals because you have the vestibule, or the semicircular ducts, telling your brain that you're spinning, but your other senses, like your eyes, are telling your brain that it's not, so it gets very confused, and that's why you run into things. Here we see that directional rotational motion causing those cupulas to bend and allowing the brain to interpret rotational motion. The utricle and saccule, the hair cells here provide sensations for position in both linear and horizontal fashions, horizontal and vertical rather, fashions. They're connected with an endolymphatic duct, which ends in the endolymphatic sac. You have some more terminology here, but I'm going to jump to an image. Here again, here's the utricle, and then here's the saccule within the vestibule. And here again are our hair cells. So the hair cells are in a gelatinous layer. We see that right here. And on top of that gelatinous layer are crystals called otoliths. It's almost like having a jello mold with a brick on top or some heavy weight on top. And that is the orientation of these hair cells. Here we see sort of an image representing that. We see the weight, the crystals on top of the gelatinous mass right here that the hair cells are embedded in. So if you were to look up all of a sudden, the weight, the otolith, the crystals, are going to be pulled towards the gravitational center of the Earth, and in doing so they're going to bend or cause that gelatinous mass to shift, and that's going to cause those hair cells to fire. And that signal that your brain gets from that action is interpreted as your head looking up, basically. So that is is how the body perceives both horizontal and vertical motion from the gravitational pull of these otoliths when motion is being felt. In terms of the pathways, sensory neurons in the vestibular ganglia, they monitor these hair cells. And the fibers are going to form the vestibular branch of the vestibular cochlear nerve. Remember that cranial nerve 8, the vestibular cochlear nerve, has two main branches. You have the vestibular nerve and the cochlear nerve. So here, since we're talking about equilibrium, it's the vestibular nerve that's carrying that sensation. That vestibular nerve is going to synapse at the vestibular nuclei, which is right between the pons and the medulla oblongata. I'll come back to this slide. I want to show you the image. So here we have the semicircular canals in the vestibule, and of course the vestibular nerve joins the cochlear to form the vestibular cochlear, which then synapses at the vestibular nucleus right between the medulla oblongata and the pons. From there, lots of signals are being sent. One, we get a signal that goes to the cerebellum. Obviously, the cerebellum assists in monitoring balance, so the cerebellum needs to know if you are accelerating in any direction. We also send signals to the cerebral cortex so that you can be made aware of the fact that you're spinning. Cranial nerves 3, 4, and 6 are going to be signaled so that they can control eye movements, so that you can try to visualize what's occurring during that spinning. And then, of course, the vestibulo-spinal tracts are going to carry signals down throughout the body to help stabilize so that you don't lose balance while spinning. So lots of signals. Back one slide, 4 functions of the vestibular nuclei. They integrate sensory information about balance and equilibrium coming from both sides of the head. So you have, again, 2 sets of semicircular canals, 2 vestibules, and the brain has to interpret from both sides. It's going to relay that information from the vestibular complex to the cerebellum for balance, to the cerebral cortex so that we're physically aware of the head positioning, and it's going to send commands to motor nuclei in the brainstem and the spinal cord to help maintain balance during times of acceleration. Eye, head, neck movements are coordinated through reflexive motor commands from this vestibular nuclei, and they're going to be distributed to the motor nuclei via the cranial nerves. So peripheral muscle tone, head, neck movements, these are all instructions descended in the vestibular spinal tract of the spinal cord. Eye movements. So anytime we have a sensation of motion, the superior colliculus of the mesencephalon is going to get involved, and its job is to attempt to keep the eyes focused on a set point. If you are spinning, again, in a chair, the mesencephalon, in an attempt to try to keep the eyes focused on a set point, causes a condition known as stagmus. And this is a jumping of the eye. If you've ever spun a child and then stopped them and looked at their eye, you can see that the eye is undergoing stagmus. It's jumping from point to point. And that's because it's just having trouble controlling eye movements caused by the sensation. If you suffer stagmus and you're not spinning, then we would look for any type of either damage to the brain stem or the inner ear. Hearing is a separate mechanism. Sound waves are converted into mechanical movements by vibrations of the tympanic membrane. When the tympanic membrane vibrates, it's going to cause the malleus to vibrate, which will then vibrate the incus, which will then vibrate the stapes. And at that point, these vibrations are converted into pressure waves that are found within fluids. of the cochlea. Remember, the stapes is connected to the cochlea. Let's look at some of the physics behind pressure waves. A wavelength is the distance between two adjacent crests, and the frequency is the number of waves per given time, per unit time. So a low frequency would look like this compared to a high frequency in which you have more waves per unit time. Usually these can be measured in terms of cycles or hertz, depending on whether it's physics or physiology. Pitch is our response to frequency. Amplitude is the height of the sound wave, and we can associate that with loudness. So there is a very loud sound wave. Here's one that's not so loud. And intensity is the amount of energy within that sound wave, and it determines how loud it seems. It's reported in terms of decibels. Some of the anatomy of the cochlea. The cochlear duct lies between two ducts. We have the vestibular and the tympanic duct. These are called the scala vestibuli and the scala tympani. Hair cells lie in spiral organs called the organs of Corti, and that rests on top of a membrane called the basilar membrane. And the basilar membrane is going to separate the cochlear duct from the scala tympani. And the hair cells, while they lack kinocilia, they do have stereocilia, which makes contact with a membrane that lies over them called the tectorial membrane. There was a lot of anatomy there, and we'll show that in just a minute. Here we see the cochlea. Here's a cross-section of the cochlea, and we can identify all of these ducts. So here's the scala vestibuli, the cochlear duct, and then down here the scala tympani. So we have these three ducts that are sort of side by side with the cochlear duct in the middle. And the cochlear duct is where we find the spiral organs that are associated with those hair cells that perceive sound. Here we see sort of a blown up image of those three structures. And then, of course, down here we actually see a yet larger image of that tectorial membrane, basilar duct, and the hair cells. So here is our basilar membrane, and on that we have our hair cells are situated. And then over the top of the hair cell is the tectorial membrane. So how does sound work? Well, again, sound waves are going to permeate through the air, and they're eventually going to hit the pinna, your ear, and they're going to be directed into the external acoustic meatus, and that eventually is going to hit off the tympanic membrane. The tympanic membrane is going to cause the malleus to vibrate, which will in turn vibrate the incus, which will in turn vibrate the stapes, which will then punch into the cochlea, very similar to you punching into a waterbed. And from there, those pressure waves are going to move throughout the fluid within the cochlea. So let's go through the notes. The auditory ossicles convert pressure fluctuation in the air into much greater pressure fluctuations in the fluid. The fluid is called a perilymph of the cochlea. The frequency of sound is determined by which part of the cochlear duct is stimulated, and I'll show you that in a minute. The intensity or volume is determined by the number of hair cells that are stimulated. From softest to loudest, we can hear a range that represents a trillion-fold increase in power. Young children have the greatest hearing range, and as we age, we accumulate damage. That damage comes in terms of the loss of flexibility within the tympanic membrane. You can get a stiffening between the auditory ossicles, or you can ossify the round window, which can also lead to problems with hearing. Here we see sort of 6 basic steps in the process of hearing. Number 1, sound waves arrive at the tympanic membrane. Tympanic membrane is going to vibrate, causing a vibration within the 3 auditory ossicles. Step 2. Step 3, the movement of the stapes at the oval window produces pressure waves in the perilymph of the scala vestibuli, and that's right there at step 3. Step 4, the pressure waves distort the basilar membrane. So here we have our 3 chambers, 1, 2, 3. And as pressure waves move down the first chamber, at some point it's going to cross. As it crosses the cochlear duct, it's going to distort the basilar membrane. Remember that membrane that lines the cochlear duct. And wherever along this path, along this little blue line, wherever that occurs, that is how frequency is perceived. So 5, vibration of the basilar membrane causes those hair cells to vibrate against the tectorial membrane, and we perceive frequency at that point. Step 6, information about the stimulation is relayed to the central nervous system via the cochlear nerve. So here we have sort of the unrolled snail shell or cochlea, if you will, and we see all 3 of these ducts. So we have 1, 2, and 3. And here we have the stapes at the oval window. So as the stapes punches into the oval window, a pressure wave is going to move through this first duct. At some point it's going to cross. Wherever it crosses, and in this case it's crossing right here, that is how frequency is perceived. So we're somewhere between 6,000 and 1,000 hertz at that point. So just a rough estimate, I would say 3,000 hertz, if you will. And that is how we perceive frequency. That sound wave is then going to move down this duct until it hits the round window, which should bulge. Here we see the round window pushed outward. And it is then going to sort of elastically rebound, and that's going to dissipate all that movement, all that energy within the cochlea. So this is how we sort of dissipate energy so that you don't have sound waves bouncing back and forth. In terms of pathway, the auditory pathway, you have afferent fibers of the sensory neurons within the spiral ganglion forming the cochlear nerve. So here is your cochlear nerve, and we have, again, sensory neurons of the spiral ganglion. All these sensory neurons form and join, create the cochlear nerve. The cochlear nerve is going to join the vestibular nerve to form the vestibular cochlear nerve, cranial nerve number 8, and that's going to go straight to the medulla oblongata, where it synapses at the cochlear nucleus. From there, information ascends via the superior olivary nuclei to the pons, as well as the inferior colliculus of the midbrain. The midbrain coordinates both unconscious, they coordinate unconscious motor responses to sound. So here we see that again. Let's sort of follow that through. So from the ear, we have the vestibular cochlear nerve carrying the information all the way to the superior olivary nuclei of the pons. We see those right here. From there, we go to the inferior colliculus right here, up to the thalamus, where the thalamus decides whether or not to send it on to the region of the cortex associated with hearing, so the auditory cortex within the temporal lobe. And that's how sound is perceived. We've already noted that from softest to loudest, we have about a trillion-fold increase in power. And we know that young children have the greatest hearing range. And this table from your textbook sort of looks at various sounds and the damages they can cause. And while I'm not going to ask anything specific, just make a note. Heavy metal rock concert, immediate danger. You are going to damage your hearing ability to some degree, and that does not repair. Gunshot, immediate danger. If you're not wearing some type of protective earmuffs during when you're shooting a gun, you will accumulate damage, and over time that accumulation really adds up. Even busy traffic or working in a noisy restaurant, if it's continuous, it will accumulate damage. So it's very important to protect your sense of hearing. They predict that your generation is going to suffer significant hearing loss compared to older generations. And a lot of that is because folks tend to buy those headphones that sit directly within your ear, and it directs music directly in. So the intensity of those earbuds really affect and damage your ability to hear, and they predict you're going to have more hearing loss than others. With age, damage accumulates. Again, the tympanic membrane becomes less flexible. You can get a stiffening between the auditory ossicles or an ossification of the round window. And as silly as this image is, it works. This is going to increase the surface area, this horn, to capture more sound waves and direct it towards the tympanic membrane. So there is something to that. And with that, we end the semester.